Jennifer Doudna was born February 19, 1964, in Washington, D.C., the daughter of Dorothy Jane Williams and Martin Kirk Doudna. Her father received his Ph.D. in English literature from the University of Michigan, and her mother, a stay-at-home parent, held a master's degree in education. When Doudna was 7 years old, the family moved to Hawaii because her father accepted a position in American literature at the University of Hawaii at Hilo. Doudna's mother earned a second master's degree in Asian history from the university and taught history at a local community college. Growing up in Hilo, Hawaii, Doudna was fascinated by the environmental beauty of the island and its exotic plants and animals. They built her sense of curiosity about how nature works, and she wanted to understand the underlying biological mechanisms. When she was in school, she developed her interest in science and mathematics. Her parents fostered a culture of intellectual pursuit in her home. Her father enjoyed reading about science and filled the home with plenty of books on popular science. When she was in the 6th grade, he gave her a copy of The Double Helix by James Watson. While she attended Hilo High School, she was influenced by Miss Wong, a chemistry teacher. She graduated from Hilo High School in 1981. Doudna entered Pomona College in Claremont, California, to study biochemistry. During her sophomore year, while taking a course in general chemistry, she questioned her own ability to pursue a career in science, and considered switching her major to French. However, her French teacher suggested she stick with science. Chemistry professors Fred Grieman and Corwin Honch at Pomona had a major impact on her. She started her first scientific research in the lab of Professor Sharon Panasenko. She earned her Bachelor of Arts degree in biochemistry in 1985. She chose Harvard Medical School for her doctoral study and earned a Ph.D. in biological chemistry and molecular pharmacology in 1989. Her Ph.D. dissertation was on a system that increased the efficiency of a self-replicating catalytic RNA and was supervised by Jack W. Shostak. From 1989 to 1991, she held research fellowships in molecular biology at the Massachusetts General Hospital and in genetics at Harvard Medical School. From 1991 to 1994, she was Lucille P. Markey Postdoctoral Scholar in Biomedical Science at the University of Colorado Boulder, where she worked with Thomas Cech. Early in her scientific career, Doudna worked to uncover the structure and biological function of RNA enzymes or ribozymes. While in the Shostak lab, Doudna re-engineered the self-splicing tetrahymena group 1 catalytic intron into a true catalytic ribozyme that copied RNA templates. Her focus was on engineering ribozymes and understanding their underlying mechanisms. However, she came to realize that not being able to see the molecular mechanisms of ribozymes was a major problem. So she went to the lab of Thomas Cech at the University of Colorado Boulder to crystallize and determine the three-dimensional structure of a ribozyme for the first time, so ribozyme structure could be compared with that of enzymes, the catalytic proteins. She started this project in the Cech lab in 1991 and finished it at Yale University in 1996. She had joined Yale's Department of Molecular Biophysics and Biochemistry as an assistant professor in 1994. At Yale, Doudna's group was able to crystallize and solve the three-dimensional structure of the catalytic core of the tetrahymena group 1 ribozyme. Initially, her group was able to grow high-quality crystals, but they struggled with the phase problem due to unspecific binding of the metal ions. One of her early graduate students and later her husband, Jamie Kate, decided to soak the crystals in the heavy metal derivative, osmium hexamine, to imitate magnesium. Using this strategy, they were able to solve the structure, the first solved ribozyme structure and the second solved folded RNA structure, the first being tRNA. They found that the osmium hexamines mimicking the normal magnesium ions were in a cluster at the center of the ribozyme and served as a core for RNA folding. This interaction created a structure in the ribozyme catalytic center that was similar to that of an active site in the hydrophobic core of a protein. Her group has crystallized other ribozymes, including the hepatitis delta virus ribozyme. This initial work to solve large RNA structures led to further structural studies on an internal ribosome entry site, iris, and protein RNA complexes such as the signal recognition particle. Doudna was promoted to the position of Henry Ford II Professor of Molecular Biophysics and Biochemistry at Yale in 2000. In 2000–2001, she was Robert Burns Woodward Visiting Professor of Chemistry at Harvard University. In 2002, she accepted a faculty position at the University of California, Berkeley, as a professor of biochemistry and molecular biology, joining her husband Jamie Cate, who was a professor there. She also gained access to the synchrotron at Lawrence Berkeley National Laboratory for her experiments with high-powered X-ray diffraction. In 2009, she took a leave of absence from Berkeley to work at Genentech to lead discovery research. She left Genentech after 2 months and returned back to Berkeley and cancelled all her obligations to study CRISPR. She is currently located at the University of California, Berkeley, where she directs the Innovative Genomics Institute, a joint center of UC Berkeley and the University of California. San Francisco holds the Li Ka-shing Chancellor's Professorship in Biomedicine and Health, and is the chair of the Chancellor's Advisor Committee on Biology. Her lab now focuses on obtaining a mechanistic understanding of biological processes involving RNA. This work is divided into 3 major areas: the CRISPR system, RNA interference, and translational control via microRNAs. In 2012, Doudna and her colleagues made a new discovery that reduces the time and work needed to edit genomic DNA. Their discovery relies on a protein named SOS9 found in the Streptococcus bacterial, CRISPR, immune system that cooperates with guide RNA and works like scissors. The protein attacks its prey, the DNA of viruses, and slices it up, preventing it from infecting the bacterium. This system was discovered by Francisco Mojica, but she and Charpentier showed for the first time that they could use different RNAs to program it to cut and edit different DNAs. In 2015, Doudna gave a TED Talk about the bioethics of using CRISPR. As CRISPR becomes increasingly used to edit multicellular organisms, Doudna continues to be called upon to speak clearly about the ethics of changing an organism's function using CRISPR. Their discovery has since been further developed by many research groups for applications ranging from fundamental cell biology, plant, and animal research to treatments for diseases including sickle cell anemia, cystic fibrosis, Huntington's disease, and HIV. Doudna and several other leading biologists called for a worldwide moratorium on any clinical application of gene editing using CRISPR. Doudna supports the usage of CRISPR in somatic gene editing, gene alterations which do not get passed to the next generation, but not germline gene editing. The CRISPR system created a new straightforward way to edit DNA, and there was a rush to patent the technique. Doudna and UC Berkeley collaborators applied for a patent, and so did a group at the Broad Institute affiliated with MIT and Harvard. Zhang at the Broad Institute had shown that CRISPR SOS9 could edit genes in cultured human cells a few months after Doudna and Charpentier published their method. Before the UC Berkeley patent application was decided, a patent was granted to the Broad investigators, and UC Berkeley filed a lawsuit against the decision. In 2017, the court decided in favor of the Broad Institute, who claimed that they had initiated the research earliest and had first applied it to human cell engineering, thus supporting editing in human cells with evidence, but that the UC Berkeley group had only suggested this application. UC Berkeley appealed on grounds that they had clearly discussed and spelled out how to do the application the Broad had pursued. In September 2018, the appeals court decided in favor of the Broad Institute's patent. Meanwhile, UC Berkeley and co-applicants' patent to cover the general technique was also granted. To further cloud the issue, in Europe the claim of the Broad Institute, to have initiated the research first, was disallowed. The rejection was due to a procedural flaw in the application involving a different set of personnel listed in the lawsuit and the patent application, leading to speculation that the UC Berkeley group would prevail in Europe. Doudna has co-founded Caribou, a company to commercialize CRISPR technology. She is also a co-founder of Scribe Therapeutics, which pioneered SOS-X, a more compact, next-generation SOS9 which can efficiently cut DNA. In addition to the CRISPR breakthrough, Doudna has discovered that the hepatitis C virus utilizes an unusual strategy to synthesize viral proteins. This work could lead to new drugs to stop infections without causing harm to the tissues of the body. In 2017, Doudna co-founded Mammoth Biosciences, a San Francisco-based bioengineering tech startup. Initial funding raised $23 million, with a Series B round of funding in 2020 raising $45 million. The business is focused on improving access to biosensing tests which address challenges across healthcare, agriculture, environmental monitoring, biodefense, and more. As Executive Director of the Innovative Genomics Institute, IGI, at UC Berkeley, Doudna is leading a COVID-19 testing center. This center processes more than 1,000 patient samples per day. Mammoth Biosciences announced a peer-reviewed validation of a rapid, CRISPR-based point-of-need COVID-19 diagnostic which is faster and less expensive than QRT-PCR-based tests. As a postdoctoral student at the University of Colorado, Doudna met Jamie Kate, then a graduate student. They worked together on the project to crystallize and determine the structure of the tetrahymena group 1 intron P4-P6 catalytic region. They married when she was teaching at Yale, and they both accepted faculty positions at UC Berkeley and moved there. He is currently a UC Berkeley professor and works on gene editing yeast to increase their cellulose fermentation for biofuel production. Their son, born 2003, likes computers and math. Doudna was a Searle Scholar and received the 1996 Beckman Young Investigators Award. In 2000, she was awarded the Alan T. Waterman Award, the National Science Foundation's highest honor that annually recognizes an outstanding researcher under the age of 35, for her structure determination of a ribozyme. In 2001, she received the Eli Lilly Award in Biological Chemistry of the American Chemical Society. In 2015, together with Emmanuelle Charpentier, she received the Breakthrough Prize in Life Sciences for her contributions to CRISPR-Cas9 genome editing technology. In 2016, together with Charpentier, Feng Zhang, Philippe Horvath and Rodolphe Beringou, she received the Canada Gairdner International Award. Also in 2016, she received the Heineken Prize for Biochemistry and Biophysics. She has also been a co-recipient of the Gruber Prize in Genetics 2015, the Tang Prize 2016, the Japan Prize 2017, and the Albany Medical Center Prize 2017. In 2018, Doudna was awarded the NAS Award in Chemical Sciences, the Pearl Meister Greengard Prize from the Rockefeller University, and a Medal of Honor from the American Cancer Society. In 2019 she received the Harvey Prize of the Technion Israel for the year 2018, jointly with Emmanuelle Charpentier and Feng Zhang, and the Louis Che Wu Prize in the category of Welfare Betterment. In 2020 she received the Wolf Prize in Medicine jointly with Emmanuelle Charpentier. Also in 2020, Doudna and Charpentier were awarded the Nobel Prize in Chemistry. She was elected to the National Academy of Sciences in 2002, the American Academy of Arts and Sciences in 2003, the National Academy of Medicine in 2010 and the National Academy of Inventors in 2014. She was elected a foreign member of the Royal Society for MEM-RS in 2016. In 2017, Doudna was awarded the Golden Plate Award of the American Academy of Achievement. In 2020, she was awarded a Guggenheim Fellowship.